All right, folks, welcome back to Unit 2, Cell Structures and Function and Cell Transport for AP Biology. In this section of the notes, we are going to cover 2.6, Membrane Transport, and 2.7, Facilitated Diffusion. And before we got to this in class, we should have done an activity with you guys moving around the back of the room, so that's what that is there. All right, so kinetic energy and diffusion. All molecules have some kinetic energy. They're moving around. They have some heat, right? Unless they're at absolute zero, they're going to be moving. Thus, since they're always moving, they move in random directions. They're not like drawn one way or the other necessarily without something else, other force acting upon them. And this has the effect of causing them to spread out, or, as you saw back in lab, having a movement from an area of relatively high concentration outwards as they're spreading into an area of low concentration. So they tend to go from high to low. Now, although each molecule moves randomly, diffusions of a population of molecules can be directional. Like I said, high to low, or across a particular membrane, for instance. If we have a membrane in the middle, it can mess with that. But generally, they go from high to low and spread out unless something else is messing with it. At the dynamic equilibrium, as many molecules cross the membrane in one direction as the other. So what we mean here is equilibrium, and it looks like everything's evenly spread out between the two areas inside the cell and outside the cell, for instance. The molecules are still moving. They're still crossing in and out. However, it appears from the outside looking in that there's no net movement, but there is still movement of molecules. They don't stop just because they reach equilibrium. So as we can see here in this diagram, on the top left, we have net diffusion. We got lots of dye on the left and more water on the right. The dye is going to, as you can see, the bouncing around is going to tend to spread out from the high to the low on the right, in the middle one, and then it's relatively evenly spread out on the far right, and we call it equilibrium, but they're still moving. Down below, same thing, it's just we got yellow and purple molecules doing the same thing, moving from high to low until they're evenly spread out and both reach equilibrium. So here we can watch this animation of diffusion.
Diffusion is the tendency for particles of any kind to spread out from where they are more concentrated to where they are less concentrated. This process can also be described as molecules moving down their concentration gradient. Diffusion across a biological membrane is called passive transport, since the cell expends no energy to move the molecules. Oxygen and carbon dioxide molecules move into and out of cells by passive transport.
So, passive versus active transport. Passive transport, as the name implies, does not require any energy. You just sit back there and passively accept things. You're sitting at your desk, at your computer right now, and you may be passively absorbing this information. You're not doing much. You're just sitting there and letting the information flow. Okay. Now, that does rely on kinetic energy of the molecules, so it won't happen at absolute zero. Materials will move from high to low concentrations, as we've already talked, they'll spread out. And this is what generally allows cells to maintain an equilibrium of concentration between the inter and exterior spaces, so within the cell and outside the cell. As long as they can get through the cell membrane, materials are going to want to be evenly spread out. And this works great for most things that your cells need. If you think about cell energy, right, and needing oxygen to carry out cellular respiration. So in your cells, you're always using up oxygen, right, in cell respiration. So you're always dropping the concentration of oxygen in your cells because you're converting into carbon dioxide and everything else, and water and things, and creating ATP. So you're using up that oxygen. You're constantly driving down that concentration of oxygen. So there's always going to be more oxygen in the blood than in your cells. So oxygen will diffuse from your blood down into your cells, down the concentration gradient from high to low. And since we're always drawing oxygen out of the blood, your blood is always going to have a lower concentration of oxygen than the air. So we're always driving via diffusion, high to low, oxygen from the air into your blood. And the reverse is true with carbon dioxide, right? We're always making carbon dioxide down here in our cells via cell respiration. So we're artificially increasing the amount of carbon dioxide. So carbon dioxide is always going to be higher in your cells than your blood, diffused from high to low, inside the cell to the blood. Higher in your blood than the air, so diffused from the blood into the air in the lungs, and we can exhale it. Most things our body uses and needs, most processes, can rely on basic old passive transport. There are some situations, though, where we need active transport. Active transport uses energy to move molecules, okay, across the membrane. That energy in the form of ATP, adenosine triphosphate, okay, and that can either be in the form of a pump protein or some other cell structures, which we'll learn about in a little bit. Often, but not always, it'll move things from low to high concentrations. So it's going against the concentrations gradients, trying to push things, so to speak, uphill in the opposite direction they would normally want to flow. That's why it takes energy. You saw that back in lab when we did our activity earlier. Okay? Not always, but usually does that. And this is what allows cells to maintain concentration gradients that differ from their surroundings. This is what allows cells to have more sodium outside than inside, or more potassium inside than outside. And that ability to do that plays a huge role in things like muscle and nerve transmissions. Okay, so let's go back to passive transport now. We'll come back to active transport later on in the lecture. Simple diffusion, okay, this is plain old regular fusion, is where we have small, nonpolar, and lipid-soluble substances, okay, that can diffuse directly through the phospholipid bilayer. Okay? If they're small enough, like we saw in that video, they can squeeze between the phospholipids. If they're nonpolar or hydrophobic or lipid soluble, all the same thing basically, right? They can diffuse through that bilayer. They're not going to get repelled by the lipids tail, so they can get on through. They don't need any help. It's very simple. Okay, that would be things like alcohol, oxygen, carbon dioxide, things of that nature. Okay, and they will go from high to low concentration, following what we've been talking about, the concentration gradient. And right now we'll switch to me working on the whiteboard and describing concentration gradients to you.
All right, so just like to talk a little bit about concentration gradients now, and I like to think of them kind of like a hill. If I draw a hill on here, all right, we got a high spot and a low spot, right? And if I were to draw a big old boulder on the top of that hill, okay, it's going to want to roll downhill, right? Gravity is going to take it. The rock doesn't need to expend any energy. It's just moving like nature intended, and it's going to go on down the hill. And this is basically like diffusion. We go from an area of high concentration, and that's what the brackets mean, concentration, to an area of low concentration. Does not take any energy to do that whatsoever. Okay, at least any energy on the part of the cell, I should say. It does take energy. The energy that's inherent to that rock right there is going to move it, and it's going to tend to move from high to low. So anytime you're given a situation where you're given the concentrations of material inside and outside the cell, you can think of it, it's just going to roll downhill. It's going to go from wherever it's highest to wherever it's lowest if it can pass through the membrane. So you can kind of think it like, a little gate right here. If it's able to pass through that, then it's going to roll downhill. Okay, it can fit through that little gate, no problem. This would be like a very small molecule passing, small nonpolar molecule passing right through the phospholipid membrane, which we called simple diffusion. Okay, on the next page, we're going to talk about something that's not simple, and we'll do that in just a minute. All right, so now we've got the same situation again. We've got a high concentration. Let's pretend it's outside the cell, and a low concentration inside the cell. And this rock wants to go in. That's great if there's no barriers, nothing there. But what if we put in a wall? It's too big to fit through the phospholipids, or it's polar, so it can't get through the phospholipids. It's not going to go anywhere, right? So simple diffusion won't work in this situation. The cell needs to give the molecule a little bit of help. And that's why we call it facilitated. Facilitated, can't spell, diffusion. The cell needs to help it out. So in a situation where the molecule is too big to fit through the phospholipids, whoa, what's going on here? Let's close that. So in a situation where the molecule is too big or too polar to fit through, polar and can't go through the phospholipids, the cell can provide help to it or facilitate it. One of the things it could do, it can provide a doorway or a tunnel. Let's call it a tunnel here, right? Looks like a rock. It's supposed to be a tunnel that rock can go through. So imagine that's just the right shape for that rock to go through, and that rock then can travel through the tunnel and on down the hill. So that's one thing that we could do. Okay, that is called facilitated diffusion. We just provide a tunnel that's just the right shape for that rock to get through. And that's one way that the cell membrane can control things. But what if we do a tunnel that's that shape? Is that rock going to be able to fit through there? No, it's going to run into it. It's going to bang up against that. It's not going to get through. So that is another way that we can do a facilitated diffusion. Those protein channels are just the right shape to let specific molecules through, just the right shape to let water through, just the right shape to let calcium ions through, for instance, or sodium ions through, and not other ones. Okay. Another thing we can do is we can do it like a door. Okay, I'm going to try my best to draw a panel door here. Okay, so we have that door, and it is big enough, let's say, to get this molecule through, but it's closed. Okay, we have something like this for glucose. We talked about glucose in the first unit when we talked about negative feedback loops and regulation. And when your blood sugar gets high, your pancreas sends out insulin, which tells your cells to suck in glucose. What it's actually doing is that insulin is kind of like a little key. That's a key that unlocks that door, and then what it will do, it will open that door, and then the glucose can come in. Sorry, I don't know how to animate an opening door. But then when the insulin is no longer being secreted, that door closes, and you keep that sugar out of the cell. And the other sugar that's coming up behind it, all that extra sugar, stays out there when you get the amount of sugar in the cell that you need, and you got the blood sugar outside the cell at the right levels. So that is also facilitated diffusion. It doesn't take the cell any energy to provide that door. Okay, all it has to do is open and close it. And that's one of the nice properties about proteins. They can change their shape. And this particular one can recognize the hormone insulin so that it knows that will cause it to change shape and let more glucose through whenever it gets unlocked. And that is also another form of facilitated diffusion. So we have regular diffusion or simple diffusion, where the molecule is able to pass right through the cell membrane, nothing to impede it. It's small and it's lipid soluble. We have facilitated diffusion where there's a tunnel that's just the right size and shape to let certain things through. And we have another type of facilitated diffusion where we have a door that we can open, open or shut to allow things in down the concentration gradients. Again, the cell's not actually moving the molecules. It's just providing a gateway to get in and out. So I hope that helps you understand this.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions such as sodium ions and hydrogen ions cannot pass through the membrane unaided.
All right, so what do we do with those molecules that we want to go from high to low concentration or reach equilibrium, but can't pass through the phospholipid bilayer? They're too big or they're polar, okay? Then we employ something called facilitated diffusion. Facilitated means to help. Like I'm facilitating your learning right now. I am helping you to learn, okay? I'm not doing the learning. You guys are doing the learning. You guys have to invest the time and the energy and the mental processes to learn the stuff, but I am here to help you, explain things you don't understand, guide you to resources that are helpful. And that's what facilitated diffusion is. Things that can't fit through the phospholipids need help. Big things like glucose or amino acids, polar things like ions. So too big or too polar, we got to give them help. These transported substances we need to get through will bind to either carrier proteins or pass through protein channels. All right, we basically need to provide them a doorway to get in. Okay, think of it like if you're in a drafty old house, right? Air can move in and out of the house pretty easily through little cracks and things in the walls. No big deal. You can't do much to keep it out. But if you want to get in, okay, you need the house to give you some help. The house provides a door or a window that you can get in. Okay. So those doors are these proteins. They provide a means for these bigger things to fit through that or normally wouldn't be able to do that. All right, so here on the top, we can see a channel protein. And they provide corridors for specific molecules or ions to cross the membrane. Once again, it is diffusion because it's going from high to low concentrations down that concentration gradient we talked about before. However, we have a protein that is the right size and shape and has the right polarity in it to allow these molecules through. And they're usually pretty specific. You don't want to just have a hole that lets everything through. That would kind of be pointless. That would be, then the cell membrane is no longer semi-permeable, it's just permeable if we got it full of big holes. So these channels are just the right size, shape, and polarity to let certain things through. One type would be aquaporins. Okay, you can think about it, water pouring in. Aquaporins facilitate the diffusion of water because water is polar, right? And so we need a way for that to get in and out easily, and every cell has those aquaporins. We also have ion channels, which facilitate the fusion of specific ions. Now down below, we can see we have carrier proteins. These are a little bit different. These proteins have a certain shape and polarity and stuff, just like the channel proteins do, to be very specific to what they let in or out. But in order to let things in or out, they change shape. Okay, you can see this first one, when the molecule comes in, when it docks with that protein then changes shape and it's able to go in. Still going down its concentration gradient, still using the energy of the molecule moving itself, okay? But we have a tighter control on how things move through. So this one really makes sure that other things can't get through. So this is often used for larger things as well, because if the hole gets too big, things can sneak through. This is a way to keep things from sneaking through. Kind of like a turnstile after you go to a Brewers game or something. All right. So here we can see kind of all these things put together. We can see the lipid bilayer and we can see the proteins in there. We can see the concentrations. For all these, we're putting the higher concentration outside and the lower concentration of all these things on the inside. On the left, we have simple diffusion, which allows small lipid-soluble, hydrophobic, nonpolar molecules directly through the phospholipid bilayer. Down the concentration gradient, no energy. That's simple diffusion. Everything else is facilitated. We got to provide some means for these things to get through. So we got a channel protein here where the red small lipid-soluble proteins are getting through. And then we have transport proteins for these bigger ones. And they're all specific to certain molecules, so only certain things get through, but still going from high to low concentration. And just wanted to remind you that for water is passively transported through aquaporins, and water is a special case, a very important case, so we actually call that osmosis. It is diffusion of water across a membrane. We call it osmosis. All right. So molecules travel down their concentration gradient. That is to say, they go from high to low concentration. And if I haven't already done the concentration gradient on the whiteboard, we'll do it right now. All right. So active transport uses energy to move solutes against the gradients, usually. Sometimes it just moves them across really with the gradients, but still needs to use some cell energy. We'll talk about that special case. So facilitated diffusion, like we talked about, is still passive transport because the solute moves down its concentration gradient from high to low, and the transport requires no energy expenditure from the cell. Obviously, the molecules themselves have energy that's causing them to move, but the cell itself doesn't have to provide any energy. Some transport proteins, however, can move their solutes against the concentration gradients, push it uphill. Okay, think like a water tower. We got water towers in town, right? Water wants to flow downhill, but what we do to get water to the top of those water towers is we turn on an electric pump. It pumps the water out of the ground and pushes it up to the top of the water tower. Uses energy to get it to go against its gradient, gets it to go high. Okay. So one very important type of these are protein pumps, and generally they cause a concentration of particles. So normally diffusion, passive diffusion, simple and facilitated, we're going for equilibrium. Okay, we get equal, it's going towards an equal concentration. Here, with protein pumps, we're generally causing a concentration of particles, the opposite, right? In this case, we're moving a whole bunch of potassium in and sodium out of the cell. I don't think the animation is going to play, but we can see it right here. We have a protein pump. Okay, we'll start with number 1. So let's get into a little bit more detail here. So in number 1, you can see the cytoplasmic sodium binds the sodium-potassium pump. On the left-hand side, it's got the right shape. Is high when the protein has this shape, so it's going to be easy for that sodium to bond. Okay. Sodium binding stimulates phosphorylation of ATP. That's dropping that third phosphorus, releasing the energy stored in that bond. Okay. And that, in turn, will lead to a change in the shape of the protein. The sodium will be released out of the cell, okay, because it changes the affinity for sodium, so it loses this attachment. See how the shape changed right there? That new shape now has a high affinity for potassium, which will bind in there, and that will trigger the release of that extra little phosphate group stuck onto the underside of it. That causes it to change shape again, back to its original shape. Potassium is released into the cell, and we can begin the cycle over again, pumping sodium out and potassium in. Let's watch this little video that shows it in more detail.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy from ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here is how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein, providing the energy that causes the protein to change shape and push the sodium ions across the membrane, where they are released outside the cell. Potassium ions now bind to the transport protein, and the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transport protein is now ready to repeat the process.
So why is this important? How is it that the ion pumps maintain membrane potential? Membrane potential is the voltage difference across a membrane. So when we're talking electricity, when we say voltage, that's actually potential. The potential of a charge to move from one place to another. Okay, the bigger the potential for that to happen, for the charge to move, the higher the voltage. Okay, so the bigger the difference in those two sides between the plus side of the battery and the negative side of the battery, the more voltage it's got, the harder it's going to be pushing electricity through a battery, let's say. Okay, in cells, we don't generate our electricity like here in the ceiling, in the computer you're watching the screen. What's generating the electricity is the movement of electrons, charged electron particles moving from one atom to the next. In living systems, what generates electricity, guys, is the movement of charged ions. Specifically in the case we're talking about here, sodium and potassium ions across the membrane. If we can get those ions moving fast enough, It is electricity, the movement of charged particles, sodium potassium ions in this case. So voltage is created by creating differences in the distribution of positive and negative ions across the membrane. This is what gets nerves and muscles that need to generate electricity. This is how we do it. Okay. So an electron pump is a transport protein that generates a voltage across the membrane. We put lots of sodium outside, lots of potassium inside. Okay. That is the major electrogenic pump of animal cells. And with this huge amount of sodium outside and the huge amount of potassium inside, if we open up an ion channel, we got a closed ion channel. If we get a signal, say from the brain or from another nerve to open up that ion channel, now all of a sudden sodium has got a ton of voltage or potential, and it's going to try to rush through to reach equilibrium, right? Because we got a huge concentration difference, a really steep concentration gradient. Sodium rushes in. That rushing into the sodium generates electricity. So this is how we do it. We spend our time in between electrical impulses, pumping sodium out, potassium in, and that way when we need to generate the electricity, all we got to do is open up a protein channel, and it rushes through, generates the electricity. It's kind of like going back to that water tower example before, right? We spend our time in between people, the fire department needing to hose down a fire, pumping water slowly to the top of the water tower, building up a huge amount of potential energy in this water way up high. Okay, that takes energy, right? And we're storing it in that water tower. When the fire department turns on their hose, they open up the tap, gives a place for that water to go. It's an open channel. It all comes out with a lot of force and a lot of speed. It's the same thing, except with ions moving across the membrane here. So here you can see that. And this is in plants. We have protein pumps that are moving hydrogen across. That's kind of their version of it. Okay. So just to summate what we've covered so far, we got passive transport on the left, simple diffusion on the far left with small nonpolar hydrophobic molecules passing directly through the phospholipids. Then we have a couple different versions of facilitated diffusion, still moving under the molecule's own energy, no energy from the cell. We got a protein channel and a carrier protein there in blue. Then on the right, we have a version of active transport. In this case, it looks like they're showing us a sodium potassium pump again that's going to use cellular energy, ATP, to move these molecules against the concentration gradient uphill, requiring energy, just like pumping water to the top of that water tower. Okay, what we haven't talked about yet is things that are too big for these protein channels and these protein pumps to move. Okay, small molecules and water enter or leave the cell through the lipid bilayer or via transport proteins, including active transport ones. Large molecules, such as polysaccharides and big proteins, cross the membrane via vesicles in something we call bulk transport. But that requires energy, and those two forms of bulk transport, the main two forms, are endocytosis and exocytosis. Cytosis means cell movement, endo means bringing things in, exo means moving things out. Now, they could be working with the concentration gradient or against it. It doesn't really matter in this case. It matters that the cell needs to use energy to do this because molecules are so big, we basically have to open up the cell membrane to bring things in or bring things out. Okay, and that requires energy. We're using those actin filaments, those microfilaments we talked about earlier on in this unit to move that cell membrane to bring big things in or out. So exocytosis, transport vessels migrate to the membrane. So here's the membrane, they migrate to the membrane. They fuse with it, hydrophobic, they fuse, and then they fuse, open up, and then the stuff comes out, and we've added a little bit of extra membrane to the surface of the cell. Okay. Many secretory cells use exocytosis to export their products, like various hormones, oils, neurotransmitters, things of that nature. Endocytosis, bringing things in. The cell takes in macromolecules by forming vesicles from the plasma membrane, so it kind of in pockets and absorbs the stuff and pushes it through. We'll show a video in a minute that'll make that a little more clear. Endocytosis is a reversal of exocytosis involving different proteins, but it's still basically the same thing. 3 types of endocytosis. There's phagocytosis. Phago means eating. Pinocytosis, which means drinking, so taking care of liquid things. And receptor mediated endocytosis. Let's take a look at each of those. All right, let's take a look at phagocytosis. In phagocytosis, the cell engulfs a particle in a vacuole. And so we can see how the cell membrane is reaching out to surround that food particle or other particle, engulfing it and bringing it in. And the vacuole fuses with the lysosome, and the lysosome can put in those digestive enzymes and break it all down.
Endocytosis is the movement of materials into a cell via membranous vesicles. Exocytosis is the movement of materials out of a cell via membranous vesicles. These processes allow patches of membrane to flow from compartment to compartment, reminding us that a cell is a dynamic structure.
In Pinocytosis, it's basically the same thing, except we're bringing in fluids and the materials dissolved in it, kind of gulped into the tiny vesicles for Pinocytosis. Here's a video of that.
In Pinocytosis, or cell drinking, the cell engulfs extracellular fluid, including molecules such as sugars and proteins. These materials enter the cell inside a vesicle. Epithelial cells in capillaries use pinocytosis to engulf the liquid portion of blood at the capillary surface. The resulting vesicles travel across the capillary cells and release their contents to surrounding tissues, while blood cells remain in the blood.
And the last one I want to discuss is receptor-mediated transport, or endocytosis. And here we've got bindings of ligands to receptors triggers vesicle formation. So basically, we've got a signal that binds specifically to a certain receptor, and it says, hey, open up and let us in. Okay, it's the lock and the key.
Unlike phagocytosis and pinocytosis, receptor-mediated endocytosis is very specific. It is triggered when membrane receptors bind to specific external molecules, such as protein cholesterol complexes or proteins bound to iron. Membrane vesicles pinch off, and the external protein and its cargo are brought into the cell.
One review video here, and then we'll call it quits.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is co-transport. Here, both sodium ions and glucose move into the cell through a co-transporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient. Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, The plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer. Cells use all these processes to get what we need.
All right, folks, that'll do it for that section of the notes. We'll cover tonicity and osmoregulation and mechanisms of transport in class later. Have a good day. Bye.